guess is this turned on? Okay. I just wondered, follow up to that last discussion by Patrick, if I could just ask you a question, Patrick. I was interested if we consider human embryos as, right here, hello? Okay, if we consider human embryos as humans, we have some problems in terms of fertility clinics, and we also have some problems in terms of the person may never have been implanted in the wall of the uterus. There never was a placenta. There was never an umbilical cord. And there was never a name given to this person. The person never took a breath of air, which humans are known to do. And if indeed each one is a separate person, what if a couple has, let's say, 50 frozen embryos? Shouldn't they get 50 IRS deductions? Shouldn't they? They're all people, right? All of these things are people. So it's hard for me to understand just the logic behind all of it. And finally, if indeed this is the case, how is this different than imposing a minority view, such as prohibition in 1919, upon the whole United States? And how is this different in terms of tying the hands of our federal government and federal funding of embryonic stem cell research? I just don't see, you know, that was a minority view that was overthrown 16 years later in 1935, you know, and how is this different from that?
Pat, maybe you could address, in particular, the in vitro fertilization technology as a treatment of infertility.
Well, in vitro fertilization technology, as it's done in the United States, is usually there are maybe 7 or 8 embryos that are created, and then one is picked and implanted, and then the others are frozen. And so now we have about 400,000 or 500,000 embryos that are frozen. There are a couple of questions you have about that. Okay, I would say those are human beings. I think they meet the definition of a human being. That they are distinct. Obviously, they're distinct. They're not parts of the mother anymore, but they're human. They have the genetic structure of a human, but also unlike, say, a human tissue, I think they have all of the information in them, and the genetic and the epigenetic composition needed to actively develop themselves toward maturity. Now, ask, okay, if they have 50 frozen embryos, my answer to that one's going to be yes, they should get 50. That's an easy one, okay? I'm going to say, okay, right, yeah. Yeah, they should get 50, okay. Well, they shouldn't have done that to begin with, okay, that's what I'm going to say. But okay, I'm going to say, okay, but more seriously, back to why is this different from, say, prohibition, okay? Because here we're asking about the question, who is a subject of rights and who is not? Where do we draw the line? Our laws protect some human beings, and they do not protect, our laws protect some beings, and they do not protect others. Where we draw that line cannot be arbitrary, and it's not just up to, if we have a vote on it, that's fine, but that vote may be right or wrong, and we have to look at reason, and have to look at logic behind that and see, okay, well, where should we draw the line as to who is a subject of rights and who is not? It's not an arbitrary decision. And I would say that every whole human being is someone who is a subject of rights. And that would be, you know, I have a long argument for that, but I won't bore you with it right now. But I think that I think any other way of drawing the line, I think is going to be an arbitrary line drawing, and also will end up denying the fundamental dignity, equal dignity of all human beings.
Would anybody in the panel like to respond to this question?
just a very brief follow-up, because in my own presentation, I kind of lost track of where I was, and this really did pick up on exactly the kind of point I wanted to make with regard to the chimera experiments. That is, if you listen to this conversation, and if this conversation were allowed to proceed for a fairly lengthy period of time, I think we'd begin to focus on the word human in the concept of human being. Because what we're hearing is an argument that is based on, at least in part, on the notion that biology helps to define categories. One of those categories is human, and that triggers a certain kind of analysis about who should be the subject of rights. And it's exactly why, for example, the chimera work, which may really challenge our notion of what is human, will play back into this debate about how it is that we use science to help in our conversations about the definite, about what should be the subject of rights. The second observation simply is that there's an interesting phenomenon, at least for me, in watching the scientists repeatedly say, you know, science can't answer these problems, right? We'll tell you, we'll tell you when a particular gene turns on and off, and we'll tell you when you've got a haploid versus a diploid cell, but that is not going to tell us whether or not we need to treat this particular cell or this particular organism in a particular way. So the scientists tend to be fairly modest about what science can do. And yet the non-scientists among us tend to constantly reach to science for what you're describing as not arbitrary, or other people define as clear or unmistakable or undebatable truths about these entities, and then equate that biological truth with some conclusion one would make about how we ought to treat it. So I do think that there's a kind of irony that the non-scientists seem to put a lot of faith in science to give us a techno-fix to these debates that otherwise force us to confront fundamental differences in how we define what it is that is important enough to draw the line between the subject of rights and not the subject of rights. And that's not an argument against you so much as an observation about some of the intricacies of the way we debate these things.
We have another question from the audience.
I think I can make enough noise.
Okay. Go right ahead, John.
Okay. I thought you did a good job. You're obviously going to get it again here. You did a good job presenting your point of view. I can't say that I agree with it, but I would like to take issue with one point, I think, that you say that as soon as the sperm and the egg unite, that's a human being or a human person. You use both those terms. And I just disagree with you so much there. A human being or a human person, not a little bunch of cells. It's somebody like you. A baby is a human being. A fetus along in time in its development, perhaps for 20, I don't know, forget the number of weeks, that isn't important, becomes something that looks like it's going to be a human being, but it's still not a human being. Human being has a functioning brain, and you can talk to it, as feelings, it feels pain, etc., etc. A little syncytial cell is not a human being, no more than an oak acorn is an oak tree. If someone calls you up and says, hey, you're going to bring the cell in for dinner, you're going to bring some tomato seeds and all that stuff, and call it a tomato, it's not the same thing.
I certainly want to let Pat respond to that, but does anybody else want to get involved in this conversation about potential? Pat?
I didn't make my point. I disagree with you saying that it's true that Dave is a human being, and that it's not a rule. I think you're enunciating a rule, not a truth. That's the point of that.
That's a good question. I think there are two levels of questioning here. One is, when is it that you have a distinct whole human being? And I think we look to the embryological data to help us solve that. That's not going to settle the issue by itself. So I'm not looking to science to settle the whole issue, but I look to science to indicate, well, at least give the data so we can settle when is it that we have a distinct being that's a human being, that's a whole human being, Now then the question is, okay, well, when is it a person, or when is it a subject? Whether every whole human being is a subject of rights, science isn't going to answer that question. I think the issue there's I'm not sure whether you're disagreeing on the question, sort of the factual question, whether this is a whole human being, or whether you're just differing on the evaluative ethical question of whether these whatever it is, whether it's a small human being, whether it's a subject of rights. But I think that it's not just a clump of cells. The embryo is a very organized, architecturally organized organism that has within it the program, the genetic and epigenetic program, that is sufficient so that when placed in the right environment, this being will actively develop himself or herself to the stage of a mature human. Now that means it's an individual member of that species. An acorn is a member of the species oak. So it's not an oak tree, because oak tree is kind of a phase sorter. We only have to call things that are mature, but it's the same kind of being. Now, so then the question is going to be, right, so I think it is a whole human being. Okay? Then the question is, well, are we valuable as subject of rights in virtue of the kind of being we are? Or are we valuable as subject of rights in virtue of some other attribute we acquire later in our life? Our life begins, I think, when this human organism begins. Then the question is, okay, well, do we become valuable as subject of rights some stage later? And I think we become valuable as soon as we come to be, that we're valuable in virtue of the kind of being we are. That fundamental kind is a kind that has an orientation to develop to the point where it can reason, make free choices, and so on. I don't want to go on and on.
I just quickly wanted to say I'm keeping quiet right now because I'll be talking about this some more tonight. So why don't you all come back? Question, and well, it's very brief because we ought to get to other people who've spoken. It's my understanding that the Roman Catholic teaching is not as defined or definitive as you present it. I thought that the Roman Catholic teaching was, we don't know for sure, but just because we don't know for sure, we don't think we should take a chance on killing a human being. So therefore, we prohibit any kind of, embryonic stem cell research, abortion, etc. Is that not the case?
Yeah, it is. I mean, the idea is that, well, it's a little different from saying we just don't know. It's saying that there's good evidence that it is, and that if you have a doubt, in other words, if you could show this is not a whole human being, then game over. Then they're in a different category. But the Catholic position is that, look, the Catholic position is that there's good evidence that this is a whole human being, and there's good evidence that it is and also a whole human being is a person, namely a thing that has the nature to be a rational being. And therefore, if I choose to kill it, I'm choosing to kill what might be a human person, and so one ought not to do that. That's the Catholic position.
I think Clive wanted to respond to this.
Yeah, I want to let the people talk as well. Well, first of all, we say tomato, you say tomato. I don't know if there's any relevance to that. A lot of this is semantics about when we decide, putting rules in the sand. But there are some very important rules that have been put in the sand in England. And we have legislation, we have laws. And I'm slightly biased, but in England, when the primitive streak starts during development of the brain, which is around 14 days, in England, you're not allowed to manipulate human embryos beyond that point, or you can be put in jail. And so there are laws and regulations, very, very thoughtful laws and regulations that have tried to put lines in the sand to where we scientists can go. Unfortunately, in America, there aren't laws. This is still National Academy of Science guidelines. They haven't sat down and debated and put laws in place to regulate this field. And I think that needs to be done through these agencies that Cynthia mentioned, not fixed laws.
Didn't the President's Council on Bioethics say that they would condone the use of human embryos in research?
But that's
after 14 days. That has no legal authority.
no legal authority. So in England, it's actually legal. So we've actually made that decision. Now, that's just to answer your question directly. There are guidelines and there are set points. So we can't just do anything with embryos, so we're running under those guidelines. The second point is, you know, these 400,000 frozen embryos will run out of funders to keep them frozen, and so they are going to be destroyed eventually. And the question is whether we adopt them or whether they put to some use. And so that's another question. But these are very complex issues.
If I could ask Dr. Odorico and Dr. Spinson a question about induced pluripotent stem cells, which seem to avoid a lot of the concerns Pat and many others have. Why shouldn't we just shift our resources over to induce pluripotent stem cell research and not continue to engage in human embryonic stem cell research? Are there scientific reasons or technical reasons why human embryonic stem cell research has got greater scientific or medical promise?
Well, I think that's a good question, and that's the obvious question after the discovery or derivation of those cells. And I addressed that a little bit, I think, in one of the slides towards the end of my talk, and that was the aspect about these may not be biologically equivalent entirely to human ES cells or have the same developmental potential or have the same therapeutic potential. So I think what the scientific community feels currently, if one could take that as a general statement, that probably research should be done with both, and that we should not necessarily throw out the research to be done or that has been done in the past on human embryonic stem cells themselves and ignore that or ignore their therapeutic or developmental potential and simply move on with IPS cells. For a variety of reasons, the IPS cells may be are genetically manipulated, so they may not have the same therapeutic potential or developmental potential that I mentioned. So we may be setting back research if we were to completely disregard all that ESL research.
Just very quickly, I agree with that. My analogy is like in the car world, it's a concept car. So IPS cells are a concept car. We don't know if they're really going to work, the wheels are going to fall off, or if they're just going to, you know, be too expensive to manufacture. We're not throwing away our Fords and Toyotas when a new concept car comes from GM. I shouldn't get political. So we have to keep hold of the technology we know and build on it. It's no good just to throw away something based on one or two new papers. So we need to keep working in all of these fields, respecting the moral and ethical issues. We need to keep working on all of these fields simultaneously. The approved cell lines, and hopefully one day the new cell lines that can be added to those.
We have time for maybe one more question from the audience.
I think I can shout too.
For television, we need the microphone.
I have a couple questions, and one is how does, and this is mostly to Dr. Lee, how does this relate to the use of the human oocyte? Is there any moral responsibility not to use parts of our reproductive system? And then the second question is, are there Catholic institutions right now, universities, et cetera, that are using the pre-existing embryonic stem cell line? And then to the scientists, is there a problem with the pre-existing line, kind of like the GM car that's too old now and not being able to use it anymore?
That's the answer to that.
Got it. Yes, the answer to the last one.
We need new cars, fast sports cars. So I agree with a lot. That's the correct answer. We need new lines, just like you need new cars. New technology comes along. They grow them in different ways. We have different ways of growing the cells from 8 years ago. So we need to derive new lines and get them going and make them available to the researchers and to the clinicians if we get ahead with the clinical trials.
And just on the clinical trials, none of the original lines could be used in clinical trials at this point because they were originally grown on non-human material and they've not been maintained all along under conditions that would meet the FDA's very strict rules for guarding against any kind of contamination, so-called good manufacturing practice and good laboratory practice rules. So if you do want to move into the laboratory, it can only be done with tissues that are being developed or cells that are being developed from newer lines.
One answer to your question, I agree with both of those statements. Another answer to your question of why we need new lines, or a reason why we need new lines, is the following. How do we learn about important aspects that control human development and how we might correct genetic defects or correct congenital defects or prevent them from happening? Well, one is to rely on genetics and development from lesser organisms. Unfortunately, it's different than humans, and we cannot extrapolate directly from those organisms to humans to learn about how human developmental defects form. So to do that, one could, instead of using directly manipulating human embryos, which I think most of us would find somewhat objectionable, one could derive human embryonic stem cell lines anew from genetically abnormal backgrounds, and then compare those to normal or wild-type human embryonic stem cells. So now you have a model system to look at how that gene or growth factor or other mechanisms work in an genetically abnormal situation to a normal situation, study control of the developmental processes, the epigenetics of developmental processes, and learn a whole lot more about human development than we already know, which is very little and is not very taught really in medical school, in fact, because of a large gap in our understanding of human development compared to lesser organism development. So by deriving new lines that are from abnormal genetic backgrounds, comparing them to normal human embryonic stem cell lines, we could learn a whole lot about human development.
On the question about ova, I think the main problem is with super ovulating, where women are asked to take something that's going to cause them to produce many ova at the same time. And I don't think it's intrinsically wrong to donate body parts that are not vital body parts, but blood or semen or ova for experimental reasons. I do want to say something about the primitive streak, which appears at about 14 days. That does seem to me to be an arbitrary line, because this embryo has the structure and has the design, as it were, to develop this primitive streak, which eventually will develop to the whole nervous system in the brain from the very beginning. And of course, it doesn't actually, this brain, or the primitive streak is the rudiment of the brain. The brain doesn't actually begin to function until 11 or 12 weeks somewhat, and it doesn't actually begin to, it hasn't developed to the point where it can give you consciousness, the conscious kind of level of consciousness that is not in other animals until even way after birth.
Somewhat surprisingly, the primitive streak is absolutely crucial to deciding some of these moral issues. It's something that we will discuss at greater length, at least in more detail, maybe not greater length, but more detail, at 7 o'clock at the UWMC Theater. I think we're going to call it an afternoon. Those of you who have decided to have dinner with us, the dining room is, I think, right upstairs. Those of you who are going to go home for a nice home-cooked meal, I hope to see you at 7 o'clock at the UWMC Theater. Thank you very much. Please give a round of applause.